Hi, I'm Josh McGrath, Soil Management Extension Specialist at the University of Kentucky in Lexington. In this presentation, we're going to talk about soil analysis. It's one of the steps in soil testing. The 4 steps of soil testing are soil sample collection, analysis, interpretation, and making recommendations. So soil sample analysis is basically a chemical analysis. There are multiple different extracts that we use for soil testing, and they're regionally specific, and they're based on correlation and calibration by typically the local land-grant university. But you can see in this example that you have, let's say phosphorus in a field or in the soil, and you have increasing amounts of availability. And these different soil chemical extractions like Malic 3 or Bray are going to take different slices, or even modified Morgan, it extracts very little. And so you can't use the recommendations developed for Malic 3 and use a soil test analysis for modified Morgan. So to understand soil test chemical analysis, we need to understand a little bit about soil nutrient storage in these different pools, we call them. And so this figure shows that we've got some mineral soil elements, we've got organic matter with some of those required elements, and then we've got the exchangeable elements like potassium on the CEC. And then we've got the soil solution. And plants can only use nutrients that are in the soil solution, in that water that's in the pore space in between soil particles. And the solid material over here is in equilibrium with that soil solution. And so as the plant takes up nutrients out of the soil solution, some of this exchangeable nutrient comes off and goes into the solution to kind of backfill and be available for the plants. But these are different kind of discrete pools, and you have this mineralization from the organic matter or dissolution from that mineral portion of the soil. And this happens at different time periods. So that CEC, that exchangeable nutrient, is available pretty immediately. Whereas organic matter takes a little time to break down and mineralize. And then you have this longer term storage in that mineral pool. But this storage is from long term to short term, and it kind of represents the buffer capacity of how the soil buffers what's in the soil solution. And that's the basis of soil fertility. With an essential element like phosphorus, for example, you're going to have these kind of pools. They're somewhat discrete. I mean, we can think about them as being discrete. They might overlap in some instances, but you're going to have that mineral phosphorus that's tied to clay, or phosphorus that's in organic forms in the organic matter, and then that phosphorus in the solution pool that's immediately available to the plants. And when you have different soil test extractions, they're going to approach these different pools in different ways. And so Malic 3 is a pretty aggressive extractant, and so it's going to take a lot out of each of these different pools, whereas something like Bray isn't as aggressive, and it's going to take less, and modified Morgan is going to take even less. So that's why you get a much lower number or value back from a modified Morgan test that's used up in the Northeast New England states versus Malic 3 that we use here in Kentucky. But the number that we get from a soil test extractant from the lab is only one part of the equation. For that number to make any sense, it really is going to require that local correlation and calibration. That I'll cover in a separate presentation. But it's basically trying to relate that soil test value to a yield response to added element. So we know that different extractants are different, but oftentimes people want to be able to kind of translate one extractant into another and say, well, I sent my soil sample to a lab that runs modified Morgan, but I want to convert that value to a Malic 3 number. Well, it doesn't work that simply. Some extractants are close enough that we can kind of do some translation between them, but many aren't. And so in this paper, for example, they took 51 different soil samples and they ran them through different labs, and they looked at the recommendations that would come back from those different states. And what's important to notice is not only is the relationship weak between these two extractants, but in fact, they determined one extractant determined some fields to need fertilizer, and those same fields, if they run through that Malic 3, were determined not to need fertilizer. So they don't even predict need in the same field the same way. And that comes back to not just the chemistry of the extraction, but also that correlation and calibration step. So in summary, soil testing requires this local correlation and calibration on a regionally specific conditions for specific crops and specific crop management practices in order to determine a recommendation. And so there's all kinds of different chemical analysis available out there. They're all pretty good. All the labs participate in laboratory exchange programs for quality assurance and quality control to make sure that if they say a soil has 10 parts per million Malic 3 phosphorus, that's what it has. But that's only part of the equation. You got to know what chemistry your lab is running and then use recommendations built around that chemistry.